Bismillahirrahmanirrahim. Bismillahirrahmanirrahim. So, dear students, so today our lecture is dominance and codominance. So, when we talk about Mendelian genetics, and then we talk and then we also talk about non-Mendelian genetics. In Mendelian genetics, there are two concepts. One is dominant, and the other is recessive review. So, in this case, we can say one allele is dominant over the other because the dominant allele can mask the recessive allele. So we can see here the genotype capital P, the phenotype is purple in the first case. In second case, so genotype is small p, and phenotype is you can see it's white. And in the third case, genotype is hybrid, so it's capital P, small p, phenotype is purple. And when we review the problem, so we can see dominant and recessive. And we can see here in pea plants, purple flowers that is with the capital P are dominant over the white flower that is small p. So the cross between the two heterozygous plants. In this case, we can see that the genotypes ratio with the capital P 25% and capital P small p is 50%. So we can see here and no, when we talk about non-Mendelian genetics, in that is incomplete dominance and then complete dominance. What is codominance? Multiple alleles, polygenic traits, sex-linked traits. Incomplete dominance, first of all. So neither allele is completely dominant over the other allele. A heterozygous phenotype that is a mixture of blending of two. So here we can see the Andal, Andal, Andalusians chickens. So incomplete dominance we can see here also neither black nor white are dominant. The offspring of a black feathered chicken and a white feathered and white feathered chicken are blue. So in incomplete dominance, a third new phenotype appears in the heterozygous condition as a blend of the dominant and recessive phenotypes. Example is dominant is when you class red with the recessive, dominant red with the recessive white. So the result is a hybrid pink. So we can see here the first is red dominant, and the last one is recessive. And then is the in between we can see smaller is white, and recessive is pink. So what is the problem with the incomplete dominance? We have to see the problem of the incomplete dominance. So we can see here the cross between the pink and the white flower. The genotypes are capital R 0%, capital R small r 50%, small r 50%. Ratio is 1:1 phenotypes. Pink 50%, white 50%, and ratio is 1:1. Codominance. In case of codominance, in the heterozygous condition, both alleles are expressed equally with no blending represented by using two different capital letters. Example is dominant black plus dominant white. You can see speckled black white phenotype. In case of sickle cell anemia, we can see the first one is capital N normal cells, capital S sickle cell, and then is some of which are the normal and sickles. In case of codominance, example in speckled chickens, we can see here the capital B black feathers and capital W white feathers, and then is a capital B is the black and white speckled feathers. So notice we can say there is no gray, no blend. Each feather is either black or white. In case of codominance, again, rhododendron, then drawn. So we can see in these flowers, allele capital R is allele for red flowers, and capital W is allele for the white flowers. Cross a homozygous pair and or the white, so we can say the red and other hairs. In case of another good example of codominance is Appaloosa horses. We can see grey horses are codominant to white horses. The Heterozygous horse, capital GW, is an Appaloosa, a white horse with the grey spots. So in this class, we can see a white horse with an Appaloosa horse. So regarding the codominance, we can see the cross between an individual with a sickle cell anemia and another who is a carrier but not sick. So there are three concepts regarding the sickle cell anemia. One is if someone is a carrier, other with but not sick, and the third one is sick. So in these intervals we can say so NS means 50% and sickle cell anemia means 50%. So you can see that normal cells 50% with the genotypes and 50% sickle cell with the genotypes. So in this case, the ratio will be 1 into 1 regarding the genotypes, and regarding the phenotypes, carrier is 50% and sickle cell is 50%. Ratio is 1 into 1 in this class. So we can say that in codominance, two equally dominant alleles are expressed at the same time. Heterozygous phenotype will have both phenotypes visible. Another good example is that short-horned cattle. So in this case, we can see there is a codominance, homozygous red capital R, homozygous white capital W. The offspring of the both and red and white hairs, and then offsprings are heterozygous and called the roan. So this is we can see here the roan horse. So another example is sickle cell anemia. So first of all, we need to understand what is the sickle cell anemia in codominance. It is basically caused by an abnormal hemoglobin, that is a protein that red blood cells use to carry oxygen. Normal hemoglobin is capital R, sickle cell shaped blood cells. People who are carriers, that is heterozygous for the disease, there is a mixture of both normal and sickle cell codominance. In codominance, neither allele are dominant, both are expressed. A cross between organism with two different phenotypes produces offspring with has both phenotypes of the parental traits shown. So we can see here both alleles contribute to the phenotype. For example, in some chickens we can see black chicken, and when you cross with a white chicken, the result is speckled chicken. So this is a good example of a speckled chicken regarding the codominance. So, so problem is with the what is the problem with the codominance? The problem is that in codominance, we can see here the cross an individual with sickle cell anemia, another is who is a carrier but not sick. The genotypes and phenotypes will be like that. Normal sick, normal cell, normal, normal cross, capital S, and then is the ratio is 1 into 1 in phenotypes, carrier 2, sickle cell 2, and the ratio is 1 into 1. So here, let's stop and think. We can say that there are two alleles for the hair color trait, red and blue. What would be the resulting phenotype of heterozygous pair if the allele showed incomplete dominance? So we can say in first case, A is red, B blue, C purple, and the D is red and blue patches. So let's say there are two alleles for the hair trait color red and blue. What would be the resulting phenotypes of the heterozygous pair if the alleles showed codominance? A red, B blue, C purple, and D is red and blue patches. Another term is multiple allele, that is multiple allelism. allelism. So when there is more than two alleles possible for a given gene, it allows the larger number of genetic and phenotypic possibilities. So blood typing. So first of all, we need to understand and we must know that there are four types of blood: blood type A, B, O, and AB. AB blood is a codominant trait, and both A blood and B blood need to be dominant in order to make a combination of codominant blood types, which is AB. So here in this class, we can see once there is a cross regarding the blood type AB. So genotype, we can see a genotype, and we can see can receive blood from the regarding the blood type AB, A, from B, AB, or OB. So as we discussed, there are four AB blood types, three alleles of the I gene. So here we can say in case of blood type A is antigen on RBC. In case of blood type B, we can say B is antigen on RBC. And then in case of I, neither A or nor B antigen. So here we can see genotype, blood type, and then antibodies. In case of blood type A, antibody that is B, and in case of blood type B, antibody is A, and in case of blood type AB, it is none, and in case of blood type A, antigen A and antigen B. So this is the cross between antigen A and antigen B. That is red, we can see here red blood cells having blood type A and blood type B. So blood type AB, and the last one is blood type O. Always remember, please, that blood type AB is a universal recipient, while blood type O is universal donor. So here is a summary. So we can see here the blood types, genotype, and then blood type A, and then blood type B, and then blood type AB, and the last one is blood type O. And then the second column, we can see red blood cells, surface proteins, that is, and then the last one is plasma antibodies. So in this case, we can see here in case of blood type AB, plasma antibodies will be B. In case of blood type B, plasma antibodies will be like A, and in case of blood type AB, plasma antibodies will be none. And in case of blood type O, plasma antibodies will be like A and B glutinal. So this is another again. We can see here blood type AB, blood type A in first column. We can see antibody antibody B, and in case of blood type B, we can see antibody A. In case of blood type AB, we can see antigen A and antigen B. Blood type O, we can see antibody B and antibody A. So this is here we can see the another very good summary showing the on the one side antigen antigen A and B, and then neither antigen A or nor AB. So in the left side we can see erythrocyte, plasma, and blood type. The last column is very interesting regarding the blood type B. Erythrocyte with the type A surface antigen and plasma with antigen B antibodies. So in and then blood type B and erythrocyte with type B surface antigen and plasma with antigen A antibodies. In case of blood type AB, so we can see erythrocyte with both A and B type surface antigens and plasma with neither antigen A nor antigen AB antibodies. In case of blood type O, erythrocyte with neither type A nor type B surface antigen but plasma with both antigen A and antigen B antibodies. So here is a summary of the codominance in multiple alleles. On the left column is blood types A, B, AB, and O, and then genotypes, and then is phenotypes. So then, sorry, Blood types can receive blood from blood type A can receive from blood type A and O, blood type B can receive from blood type B and O, blood type AB can receive from AB and O, and then blood type O can receive from blood type O. So here we must remember that human blood is a type of example of both codominance and a trait with the multiple alleles. Always remember blood type AB is universal acceptor, while blood type O is the universal donor. So this is RH factor. This is RH factor. So its name get from the experiments conducted in 1937 by scientists Carl Landsteiner and Alexander S. Wiener. So in this factor, so basically it is an antigen or more specifically a protein that exists on the surface of the red blood cells. If a person has either two plus genes for H or one plus and one minus Rh gene, they will test Rh positive and person will be negative only if they have two negative. So this shows the relevance of the Rh factor and AB blood typing. So the next concept is multifactorial. In this case, we can see that it is referring to control expression of a trait by several genes and environmental factors. Many multifactorial traits show continuous distribution. So, very good example regarding the multifactorial is the human height. Usually, there is a height between 120 centimeter and 200 centimeters. However, these genes cannot be fully expressed without all the necessary nutrients from the healthy diet. So, it means in multifactorial, regarding the human height, diet is equally very important. So, this is today we covered. So, what are the blood types, and then what are the what is the definition of dominance, codominance, and with examples. So, dear students, today we finished the lecture of the dominance and codominance.